So today we start Unit 10. Mendel and meiosis. Meiosis, hey, that sounds a lot like, oops. That sounds a lot like something we've already learned, doesn't it? And I'm not even saying it correctly. I always emphasize the me part so you guys as students can kind of make sure you know the difference. But really, it's meiosis, which, boy, sounds a lot like mitosis. And it is a lot like mitosis, unfortunately. So it's gonna be interesting. We get through, we're not there yet. We'll get to that next week. But got to make sure we learn the difference between meiosis and mitosis. They're very similar, but there are definitely differences. And again, it'd be important to learn because everybody coming back 100% brings us one step closer to coming back to tests, right? Yeah. We'd start testing again once we get everybody back.
Yeah, but some people still don't want.
It's true. We're only getting one step closer. Not all the way there, just one step closer. All right, today we are taking a look at Mendals Laws, or at least the first two. We had three of them that he came up with, and today we're going to take a look at two of the three. So Mendel is the last name of an Austrian monk. His first name is Gregor. An Austrian monk. So what does that mean? Well, it means he lived in Austria, over there in Europe, and he was a monk. So he kind of, I don't know, lived by himself, or at least with a small group of other monks, and he kind of lived in a castle, and he grew pea plants, a monastery, like a church, but I don't know, I thought it looked like a castle. But he grew pea plants, like peas, like what you eat, peas. He grew them, and he grew them for years and years and years. And not only did he grow them, but he also bred them. Yes, he crossed them, like a male and a female. Yes, plants can be male and female parts. So he crossed them. He recorded the observations. His data is outstanding. He is a model of how to collect and interpret data in the world of science. So his data is just amazing. And because of his data, we consider him the father of modern genetics. What he figured out led to our understanding of genetics. All right, Judah, you had a question?
Yeah, I was gonna ask you.
The P dude, yep. He is the P dude. Yep. He could probably tell you a million different recipes for peas. Like Forrest Gump. You guys ever seen Forrest Gump? When Bubba has like a million different recipes for shrimp. All right, another lost reference. All right. Anyway, well, I don't want to say his first law. Do not want to call it his first law because there's not really an order to these. But the first law that we will discuss is the law of dominance. So when an organism has different alleles, guys, there's going to be a lot of key terms in this unit. I think there's probably a lot of key terms in biology in general. It's like learning a foreign language. Well, this unit especially. So when an organism has different alleles, first of all, we call that heterozygous. Heterozygous means different alleles. One allele, which we call the dominant allele, is displayed over the other allele, which we call the recessive allele. You have two colors? Doesn't really matter which two, just two different colors. Make sure they're sharpened too. Let's do some awesome artistry. It's a flower. Like I said, awesome artistry. The genotype, as kind of the word sort of implies, geno, it's the genes. We'll call this one RR, big R, capital R, capital R. The phenotype is the physical characteristic. So physical characteristic pH, phenotype pH. This flower physically is red, so that's its phenotype. We call this homozygous dominant. So homozygous means two of the same. Big R, two of the same, they're both dominant. Another term for this, I'm actually going to kind of put it in the middle because this term is going to apply to both of these. But another term for this is purebred. Purebred. Versus a blue flower. Hey, this is big. I would say this is the most common mistake people make in this unit. We used big R for red. So guess what? We use little r for blue. People always try to use B for blue or whatever trait they're working with. Like let's say you guys did black and white. You try to use B for black and W for white. And you can't do that for dominant recessive. The reason why you can't do that is because we actually will do that next chapter. We're going to talk about more advanced genetics, and there is actually a time for more advanced genetics. There is a time to use two different letters, and it means something completely different. So for right now, just regular old dominant recessive, make sure you guys are using capital letter, lowercase letter. Same letter though, that's key, same letter. The phenotype here, blue, and we call this homozygous recessive. So two of the same, but they're recessive, they're the little ones. And that's why we've got purebred kind of in the middle here. Both of these are considered purebreds. Got a purebred red and a purebred blue. So what happens when we have one big and one little letter? Aha. Well, here's Mendels' law of dominance. One allele is displayed over the other. That's the law of dominance. I mean, pretty easy if you're homozygous, but yeah, if you have a red gene and red gene, you're going to be red. Blue gene or blue gene, you'll be blue. What if you have one of each? Well, one of them then is dominant to the other. Your phenotype is red. You're now called heterozygous. So homo means the same, hetero means different. Same, different. These guys are sometimes called hybrid. You know, like we're doing hybrid learning right now. Sometimes in the classroom, sometimes at home. Hybrid, one of each. Cool. Questions so far? Onward and upward. Ready for the next law? Cool. All right, the next law we're doing, I don't want to call it the second law or anything. There's no particular order of these, but the next law we're doing it's called the law of segregation. Now, this law actually has a couple of different points to it. First point, every individual. Now, when I say that, I mean every eukaryotic individual. Prokaryotes don't have this, but eukaryotes, it's the cells we're aware of. Every individual has two alleles for each trait. Okay, take a quick break here and discuss what these terms mean. It's like the third time you've seen alleles, now you've seen traits. Well, alleles are the options that a trait can be. A trait is the expression of a gene. Man, another term. Keep throwing these terms out. A gene is actually a lot simpler than what people always seem to think it is or imply that it is with their speech. And that's one of the things that always drives me bonkers. And people misuse this term all the time. And the way that they're using it is just enough to let you know that they know just enough to know that they don't know what they know. A gene is pretty simply gene is just the DNA. It's a region of the DNA that codes for a trait. So when we refer to a gene, all we're talking about is an area of DNA. Always drives me crazy when you say, oh, I've got the gene for blue eyes. No, you don't. You have misused that term. No one has the gene for blue eyes. We all have the same gene for eye color. Oops. All right. So a gene, for example, is just a stretch of DNA, and we all have the same genes. We do. We're all human. We all have the same genes. We do. We do. What's different isn't the traits. The trait is just how the gene is expressed. So for example, up here, the trait was what color of flower. The gene was the stretch of DNA. The trait was what color of flower. And finally, here's what people mean to say when they say gene. The allele. So again, it drives me batty when people say I have the gene for blue eyes, because you don't. We all have the same gene for eye color. What might be different about your eyes is perhaps you have the allele for blue eyes. But again, whenever you say you have the gene for blue eyes, what you're implying is that there is a different gene for brown eyes, different gene for green eyes, different gene for all these different options. And it really isn't. They're all the same gene. The gene is for eye color. The difference is what allele you have. What option do you have for that gene, for that trait? So that's what you mean to say when we're talking about all your alleles. I'm sorry, when you're talking about all your genes, what you mean to be talking about are all your alleles. What options for those genes do you have? Questions? Cool. All right, so it's kind of a little aside. We've got more points at the law of segregation. So like I said, we had that first point. There's several points to this one. We just kind of did a quick pause there to make sure that you guys are good with the terminology. Gene, allele, trait. All right, here's the next part, next point to the law of segregation. You have two of every allele. Two of every allele. The dominant one is displayed. How did you get two of every allele? Well, you inherited one allele from your mom and one allele from your dad. That's where your alleles came from. That's why you have a pair of them. And the next point comes right from that. kind of right along with that, you have one allele from your mom, one allele from your dad, but when you go to reproduce, you're only going to pass one of those two alleles on. Your offspring will also have two alleles, but only one of them came from you. It would be your partner who would give the other allele. So that means perhaps you have the most pretty eyes, and you got that allele from your dad. Oh, lucky you with those gorgeous, gorgeous eyes. But perhaps you also carry the ugly eye allele, and you receive that from mom. Now, lucky you, pretty eyes is dominant to ugly eyes. So you have the prettiest eyes. Congratulations. That doesn't necessarily mean you're passing on pretty eyes. Just because you have pretty eyes doesn't mean you pass it on. You may pass on the ugly eye allele that you have from mom. So dominant and recessive doesn't dictate anything as to which way it gets passed on. It's not like the dominant gene is like, oh, I'm dominant, so I'm the one that's going to get passed. Nope, doesn't work like that. Doesn't work like that. It is a 50-50 chance to pass on either allele.
We're gonna do it this time, then. Right?
Either one step ahead or one step behind. Either way, you are lock and step with me. Yep. Cool. So what that means is when we do these crosses. So for example, if we were to cross two hybrids, well, there's a lot of possible outcomes we could have, right? Because both parents, here's parent one, here's parent two, both parents have two options for what they pass on. So for instance here, first parent could pass on big R, and second parent could pass on big R, right? Or first parent could pass on big R, and second parent could pass on little r. Or first parent could pass on little r, second parent could pass on big R. Hey, by the way, list your dominant allele first. So you saw, normally I was doing first parent first, and then, well, always list the capital letter first. Get in the habit of doing that. List your capital letter first. All right. Or finally, we can also have the option of little r, little r. Whew, all right. Well, that gets messy, doesn't it? That gets messy. Luckily for us, there's an easier way. So luckily, there's a much easier way to show our potential outcomes from a cross, and that is called a Punnett square, named after the guy who invented them. Named after a guy named Reginald Punnett. Oops, there's no E on the end of that. Sorry about that. He's not French. So named after a guy named Reginald Punnett around 1910. I like to point that out because guys, Gregor Mendel, He did his work around 1860. Just as kind of a historical reference there, think about what was going on here in America in 1860. We were fighting our American Civil War. So it was around the time that our Civil War was occurring that Gregor Mendel did all the work with the pea plants and figured out the basics of modern genetics. Well, it wasn't until 50 years later that Punnett came around and invented this much easier way to do crosses. Again, this gets kind of confusing, especially when you're working with multiple traits. We'll see that later next week. You know, right now it's not terrible, but can be a lot more straightforward. So how a Punnett square works is it's a box, and you divide it into fourths. And then on the top, you put one parent, the genotype of one parent. Down the side, you put the genotype of the other parent. I'll put mom up here and dad right here, though honestly it doesn't matter. If you put mom on the side and dad on the top, doesn't matter. But one parent on top, one parent on the side. How the Punnett square works then? This big R drops. So right there and right there. The little r does the same thing. I bring the little r down to this box and the little r to that box. Again, notice, I know it's recessive. I know it's a lowercase, so I'm putting it over to the right so I can write my dominant allele, if I have one, to the left. If I have another recessive allele, big deal. Cool, then we do the same thing coming across the box. This big R comes here, and another big R comes across to right there. Same thing down here then, the little r comes across and the little r comes across. Cool. This makes it a lot easier to analyze your crosses, a lot easier than this. Again, maybe not terribly easier than right now, but it will be a lot easier when we start doing more than one trait, just kind of seeing the box. And then kind of the nice thing is that there's only 4 squares. We can all do math with 4 squares. It's pretty easy. We'd expect homozygous dominant, a purebred dominant, 25% of the time, 1 in 4 tries would be homozygous dominant, big R, big R. Heterozygous, big R, little r. That's here and here. That's 2 out of 4. Like I said, we can all do the math for 4 boxes. That's 50%. And little r, homozygous recessive, 1 out of 4, and that is 25%. Hey, that's a little bit different than the phenotypes. Because remember, the heterozygous phenotype is going to display, or the heterozygous genotype is going to display the dominant phenotype. So 50% plus 75%, or for just to count, 3 of them have at least 1 big R. 3 out of 4 is 75% red. Only 1 of them is going to be homozygous recessive. 1 out of 4, 25% chance to have a blue flower. Again, that's the numbers that Mendel kept getting. He crossed flowers. He did 100 of them. 75 of them came up dominant. 25% came up recessive. He was able to figure all this out. What's super impressive, at least I think what's super impressive, is that he figured all this out before he knew anything about DNA. DNA was unknown during Mendel's time. especially how it worked. We didn't even figure out how DNA worked and that it is the genetic material until like 1950s. 1950s, 100 years after Mendel before we really figured out how it is DNA works. Today we call the pairs of chromosomes. Again, he didn't know about chromosomes, Mendel. But today we know, again, you have two alleles because you have two of each chromosome. And we call those chromosomes, those pairs of chromosomes, homologous pairs.
Mr. Richardson, will we be writing a lot for the chromosome so I know if I need to go to the next page or not?
We are almost done, Yash. We have like 2 more sentences to put down.
Okay.
We'll finish in this tiny little area and still have a little bit of room. Yeah, we are almost done. We're nearly there. All right, so pairs of chromosomes are called homologous pairs. Homologous pairs. Humans, we have 23 pairs of chromosomes. 23 pairs, that's 46 total. Pairs 2, so 23 times 2, 46 total chromosomes, 23 pairs. Again, why a pair? Because you have one from mom and one from dad. One from mom, one from dad. Here's a big thing. I've already said it once. I like to hammer it home. Guys, we all have the same genes. We're all human. We all have the same genes. A gene is just a region of DNA. This stretch right there, we'll say. We'll call it the gene for hair color. Again, notice I didn't say the gene for blonde hair, the gene for brown hair, the gene for black hair, any of that. I said the gene for hair color. We all have it. We all have the gene for hair color.
So like humans all have the same genes, but like with like all things have the same genes, so like where humans can like dogs that have the same genes.
Very good question. So I don't know if you guys at home heard the question. The question was, okay, as humans, we all have the same genes. Does that mean we have the same genes as other organisms? And the answer to that is sort of. We do have a lot of the same genes as dogs. Yeah. We have even more of the same genes as monkeys. We have a ton. I think like 98% of our DNA is the exact same as a monkey.
Oh, there's more than that.
Not as much. Next question then. So yeah, we're really, really similar to monkeys. We're pretty similar to pigs and cows. I don't know if you guys are, you guys are like, have you guys studied like the trees of life where you like genus, species, family, all that stuff? Order, kingdoms. You've heard of this stuff before? Well, that's all molecular now, meaning that's all based on DNA sequences. We've actually rearranged. We've said these two animals are more closely related than we used to think based on their DNA sequence because they share so much DNA. So we share something like 98% of the genes with a monkey. Do they have some things that are different from us? Yes. Do we have some things different from them? Yes. But we share 98%. A banana is more like 50%, I think. So that's still a lot. Half of our genes a banana has. A lot of them we've already talked about. Think about it. DNA polymerase. Remember that guy, that protein? Where do proteins come from? Proteins come from genes. So DNA polymerase, the guy that copies DNA. RNA polymerase, helicase, the protein that opens up the DNA. These are genes that pretty much every organism on Earth has. We all have the gene for DNA helicase. We all open up our DNA, untwist it the same way. So those are a lot of the genes you share with a banana. Obviously, a banana doesn't have a gene for hair color. That would be interesting. In a couple chapters, though, we'll talk about giving a banana the gene for hair color. Genetic modifications. We're actually getting really good at that nowadays. Grapes that taste like cotton candy. Oh, I've never had them, and I don't like cotton candy, so that sounds awful, but we can do it. We can put genes into other organisms. Okay, I've got a comma there. One last line. Again, I like to harp and harp and harp about the difference between alleles and genes. Please don't ever tell people you've got the gene for blonde hair. You don't. You don't. What you might have is the allele for blonde hair. Or the allele for black hair. Or the allele for brown hair. All those other options. Alleles are the options that your genes can be. But we all have the gene for hair. We all do. We all do. Questions? People at home? Okay, very good. Guys, only about half of you have submitted 9.5, homework 9.5. The one that was assigned Tuesday. It's not due till tonight, but I'm just giving you that heads up. You had a homework due tonight. Only about half of you have actually submitted it. So if you haven't done that yet, get on that, submit it. There's a new homework in 10.1. I just assigned it earlier this morning. Goes with the stuff we just took a look at. So 10.1 is due tomorrow night. So you guys have 2 homeworks due before the end of the week here.